Hey guys, how's it going? Anybody hear me?
It's good. Yeah, I can hear you.
Okay, cool. Thanks. Thanks for the feedback. All right. Sorry, I'm running a little late for my last class. Okay. Before we start, are there any questions over what we talked about when we were talking about water relations? Anybody? Okay, then today what we're going to start on is we're going to start talking about temperature and thermoregulation. And what I wanted us to do was I wanted us to read Holden Bogart as kind of an introduction to this set of ideas having to do with reptile thermoregulation. And so I'll guide the discussion through the paper, but obviously, hopefully you guys have all read it and have some insights that I will ask you about. So this was a paper that was published, what, in 1944, I think? So in the grand scheme of things, quite some time ago, yeah, 1944, quite some time ago. And when we were going through the history of herpetology section, I mentioned that this was kind of a hallmark paper in that it's one of the first places where people are kind of systematically thinking about amphibians and reptiles in terms of their biology and how they're coping with the environments that they live in. And these guys focused on desert reptiles for a variety of reasons. What's one of the primary reasons that they were focusing on reptiles in the deserts of the desert southwest of the United States? Today's the day everybody turns off their cameras and goes and does something else during class and nobody answers me. It's the day I feared. Oh no.
So I think it's.
Go ahead.
So I think it was having to do because the desert has really high temperatures, but also like drastic temperature changes. So it should amplify any of the behaviors or like going on in the reptiles to deal with those temperature changes.
Yeah, so if you were working in the tropics where temperature doesn't fluctuate all that much, both seasonally and over the course of a day in reality, you might not see organisms engaging in a lot of thermoregulatory behaviors and things like this, simply because they live in a relatively constant sort of environment. Deserts are well known for not only wide fluctuations throughout the year. Deserts can be very cold in the winter and extremely hot in the summer, but you can also experience daily changes in temperature that go from 90 degrees or higher during the daytime to below freezing at night. And the reason for this is that the atmosphere in deserts doesn't hold a lot of water. That's the whole point. That's the whole reason that there is a desert there, is that there's very little water available, both in groundwater but also in the atmosphere. And so without that water moisture in the atmosphere, heat dissipates really quickly as soon as the sun is gone. And so you can have wide swings in temperature over the course of a single day. So that was certainly one of the reasons that they were concentrating on desert lizards. What's another reason that they were concentrating on desert lizards? In the literature review section of the paper, they talk about a lot of previous research that had been done on trying to get a handle as to what the limits of reptiles were in terms of their thermal limits. What were some of the things that they said about the thermal limits of other reptiles that had been studied prior to them doing this study?
Didn't they talk about like field studies and laboratory experiments that were basically necessary to understand basic phases of the thermal problem in the reptiles?
So there is that. But what had been published, for example, in the past about agamid lizards in the Sahara Desert, the one that I'm thinking of in particular is Agamestelio. It's on page 268.
I think it said it had really high, it could withstand really high body temperatures. It says it was like 45 degrees Celsius, which is insane.
Yeah, amazingly high body temperatures, like body temperatures that strain credibility. And so measuring temperature is tricky when you're measuring on a live organism. We have way better ways of doing it now than they had back in the 1940s still, but even before that, there were some published reports of the critical thermal maximum and the lethal maximum temperatures for organisms that were just way higher than had ever been recorded for any multicellular organism. Now, what's the thing with unicellular organisms? What do you know about the temperature tolerances of bacteria, for example? This is not in the paper. This is something you might have picked up in another class, like a genetics class.
Some of them can, oh, sorry.
Isn't it a pretty small range? Sorry, Elizabeth.
What's that, Kelsey?
Isn't it a pretty small range that they can tolerate?
Actually, among the bacteria, there's some bacteria that are well known for being able to tolerate amazingly high temperatures. Have any of you guys ever done PCR?
Yeah, but it didn't work.
Okay. All right. When you were doing PCR and it didn't work, what type of polymerase did you use? Do you recall what kind of polymerase?
No, but I could go find it. I don't remember right now.
Tag polymerase. Does that ring a bell? Okay. So when you're doing PCR, this is another one of those diversions where I'm getting you around to something by making you think about things that you already know. When you do PCR, what does the typical PCR cycle look like? Like when you program your cycles and you repeat the cycle however many times you want, but each cycle has a series of temperature changes, what does it look like typically? Elizabeth went to get her lab notes. Do you guys remember at all?
Yeah, you have to heat it up, denature it, so you like unwinding the
Separate
DNA.
two strands of DNA by heat because they're only held together with hydrogen bonds. So you use heat to split them apart, and then that gives the polymerase an opportunity to go and add nucleotides to the open molecule of DNA. Well, this is accomplished because there is a bacterium called Thermus aquaticus, which was found in hot springs in Yellowstone National Park, actually, that can withstand the temperatures of hot springs in Yellowstone National Park. And as a result of it being able to sustain those temperatures, it also has all the cellular machinery to withstand those temperatures, so that when you need a polymerase, a DNA polymerase, that will operate at high temperatures because you have to heat up your sample to get your DNA molecule to have the two strands dissociate from one another. You need a DNA polymerase that is capable of withstanding those high temperatures. So all throughout this paper, they keep talking about these are extremely high temperatures for metazoans, and this is for multicellular organisms. That's what a metazoan is. And the reason that they keep saying that is because they knew even back then that there are single-celled creatures that can withstand hugely high temperatures basically close to boiling or higher than boiling water. And so Thermus aquaticus is one of these unicellular organisms that lives in hot springs that you wouldn't even want to touch the water in it because you would be severely burned. But they function just fine. As a result, all their enzymes function just fine. And so molecular biologists have co-opted some of these enzymes to do things like molecular biology, and that's what TAC of TAC polymerase stands for, is Thermus aquaticus. So, a reminder about molecular biology. So, while high temperatures may not be unusual for bacteria to stand, they're very unusual for multicellular organisms to withstand. And certainly a gamma stelio is one of these that has had a published thermal limit, lethal limit that was much higher than seemed reasonable. Have any of you guys ever been out in 120-degree weather? 120-degree Fahrenheit weather? Kelsey, where were you at?
When I was in Spain, I studied abroad, and the last week that I was there, there was like record-setting heat wave in Europe, and it was like 115 degrees, and they don't have air conditioning there, and it was miserable. It was a dry heat, but it was so hot.
Where were you?
Doesn't make a difference at that point.
Where exactly in Spain were you?
Southern Spain, Granada.
Oh, Granada. Oh, yeah, I've been to Granada before. Yeah, it is a dry heat, but dry heat doesn't really matter because I experienced like 120-degree heat in Texas when I was growing up as a kid. When it's 120 degrees out, it's just hot.
Yeah, once it gets to that point, it doesn't matter.
Yeah, it just really doesn't matter. It's just way too hot to be outside. So yeah, once you get above a certain level, it just becomes very difficult for organisms to survive, whether you're a human or whether you're a lizard. And 45 degrees centigrade is around that temperature. And so reporting temperatures of animals out being happy and functioning under those conditions seems to strain credibility. So they then go on and define a set of definitions that they're going to use, and these are found on page, where are they found? Oh, it's on page 277, when it's in the section called criteria used in recording temperatures. So the lethal minimum, the critical minimum, the voluntary minimum, the basking range, normal activity range. What are they trying to do with these different levels or criteria?
try and set like clear definitions so that everyone's like on the same page, like control it.
Okay. So can you guys see the board clearly? So if we were drawing the rate of reaction of an enzyme, for example, and I realize you guys are probably thinking, why is he going back to all this cell molecular biology stuff or chemistry stuff? What you would basically find is that you would, at cold temperatures, the rate of reaction is really slow. Once again, this is the rate of reaction of an enzymatic reaction, a reaction that is governed by enzymes. You would find that it starts out really slow at really cold temperatures, and it gradually increases. But then once you get beyond a certain point, the rate of reaction will increase pretty quickly, and then it will plateau, and then it will drop off precipitously as you get hotter and hotter. And this looks familiar to you from some previous class that you've had. Okay, so explain to me what's happening during this curve of the relationship between temperature and the rate of reaction of an enzymatic reaction.
You're moving, as the temperature increases, you're moving towards the optimal temperature of the organism. And so the enzyme gets more efficient, but then once it gets too hot, enzymes denature in the heat, so then it's just not functional.
Okay, so in this case, you're actually getting higher rates of reaction. That's a good thing because the molecules are moving around faster. The faster the molecules move around, the more opportunities you get for enzymes to come into contact with substrate. They contact one another more often. The more often they contact with one another, the more often they fit together, et cetera, until you get up to some range of temperatures in which this enzyme is performing optimally. But then once you get to a point, the performance falls down really quickly because you're at that point where enzymes begin to denature. And what's weird is that if you want to change the y-axis of this diagram, take a lizard, put it in a racetrack, run it down the racetrack at different temperatures. What you will find with a lizard in a racetrack is it does the same thing. As you're heating it up when it's cold, you get this kind of exponential increase in the speed of the lizard. Then there is a point where the lizard is kind of at its physiologically optimal temperature. And then once you get it too high above that physiologically optimal temperature, the performance falls off precipitously. And the reason is that a lizard is just a big bag of chemicals. One way of thinking about a lizard is that it is just a big bag of chemicals. It's just a big bag of chemical reactions. And those chemical reactions are influenced by the temperature at which the enzymes that drive those reactions are functioning. So, obviously, this would identify the lethal limit for a lizard species if this was a curve of, for example, sprint speed. A dead lizard has a speed of 0. However, before you get to death, you get to some point where the lizard is so hot that it doesn't really have control over its body functions. And that represents a point where if you're a lizard laying out in the wild, you're effectively dead, because if you can't locomote to get out of the sunlight and move into the shade, or locomote to get out of the sunlight and go underneath a rock or something, you're just out there in the sunlight cooking, and you will be dead if you're not dead yet. And so there comes some point before that at which the lizard starts to lose its abilities to function, that represents the critical thermal maximum, which is the point at which you're so hot that you can't remove yourself from whatever it is that's threatening you at the time, which in this case is you're getting too hot. And so this would be what number 7 is, the critical maximum. This appropriately can be termed the potential lethal and may be defined as the thermal point at which locomotor activity becomes disorganized and the animal loses its ability to escape death, to escape from conditions that will promptly lead to its death. And so do you guys know how we ascertain what the critical thermal maximum and minimum is nowadays? So what you do is you put an animal in a container that you can control the temperature of, and you slowly ramp the temperature up, and periodically you'll go and you'll flip the lizard or the snake over onto its back, and then you wait for it to right itself and to flip itself back over on its belly. And so you just keep doing this over and over again. And when you get to the temperature at which when you flip the lizard over on its back, it's incapable of flipping itself back over onto its belly, we call this the loss of the righting response. It's when the animal has gotten physiologically to the point that if you flip it over on its back, which is not the way a lizard wants to live, it's incapable of flipping itself back over onto its belly. And that corresponds pretty closely to how they describe the critical maximum. Same thing with the critical minimum. There comes a point at the cold end where the animal gets so cold that it can't do that. In either of those cases, what happens if you either cool the animal down if you're close to the critical thermal maximum, or you warm the animal up if you're close to the critical thermal minimum? What happens to the animal? You have a really hot animal, it's incapable of righting itself, but you pick it up and you move it into the shade, allow it to cool off. What's going to be the response of that animal?
Cooler body temperature?
What's that?
Just a cooler body temperature?
It will get a cooler body temperature, but what will it start doing also?
Like gain the righting response?
Yeah, it'll go back to functioning normally. Same thing. If you have a cold lizard that is down there at the critical thermal minimum, even though it may be incapable of righting itself, as soon as you warm it up a couple of degrees, it regains its righting response and begins to locomote normally, et cetera, et cetera. If you have an individual at its lethal limit, cooling it down is not going to allow it to recover. And so that's kind of the difference between the lethal limit and the critical thermal limit, critical thermal max and min, is that this animal here can recover, an animal down here cannot recover. So if you took this animal and warmed it up, it wouldn't regain its abilities because it was just too cold for too long. The boundary at the upper end of the temperature scale is much more absolute than the boundary at the lower end of the temperature scale. You can be cold for quite some time, and as long as you eventually warm up, you'll fully recover. You can't be too hot for very long at all and then be unable to recover. So the hot end is a steeper decline, and also your possibilities of recovering, the longer you stay at those really high temperatures, the less likely it is that you are to recover. So we typically nowadays, we don't use all 8 of these different categories in thermal biology. We generally talk about the critical thermal minimum and the critical thermal maximum, because ecologically, these things are the things that really, really matter. I'm going to share my screen real quick with you. So this was back in last year, I guess. Although I thought it was earlier than that. There was a cold spell in Florida, and people were warned about lizards falling out of trees. So in Florida, there are iguanas that are an introduced species in Florida, and there are tropical species that is in Florida, and Florida is kind of a subtropical environment, but is prone to cold snaps periodically. And what was happening is these lizards were out in nature doing their thing, and then this cold snap came along, and they lost their ability to right themselves. They lost their ability to cling to the trees, which is where they normally occur. And iguanas can be pretty heavy in reality. And if an iguana fell on your head, it could actually hurt. And so these animals weren't dead. They were just down here at their critical thermal minimum. When you're lying on the ground with your belly up and it's cold out, you're essentially defenseless. If you're in an environment where there's a predator who would like to feed on a lizard, you're not going to last very long in this condition, because you are unable to defend yourself. You're unable to run up into a tree or run into a burrow to protect yourself from a predatory attack. And so this kind of represents a situation where you're at a temperature where you're physiologically capable to avoid death. And as a result, this is what we think of as the critical thermal minimum or the critical thermal maximum. And if what happened was, as these animals were doing this when it cooled down, as soon as it warmed back up above their critical thermal minimum, they were perfectly fine, and they flipped themselves over and hopped up and ran back up the tree that they had just fallen out of 2 hours or 3 hours earlier. So the reason that we think of the critical thermal minimum and critical thermal maximum is because those represent ecologically important markers on this temperature scale. Seldom do we record lethal temperatures because why? They talk about the reasons why determining lethal temperatures is hard to do in this paper.
Body temperature rises rapidly at like a certain point, and then you can like still have signs of life even after like fatal damage. So it's hard to determine.
Yeah, so it's hard to determine where exactly in that ramping up of temperature did the animal actually die. And so a critical thermal min and max is much more accurate in that there's a clear physiological response that's easy to measure, easy to see. And so you don't even see people doing lethal temperatures anymore. For one thing, institutional animal care and use committees don't really like to approve you heating your animals up until they die or cooling them down until they die. And in the end, it's not really a very ecologically relevant variable to begin with. And it's also one that is fraught with lots of error associated with it. And so we don't even really measure it anymore. This pisses off some people, some of the older herpetologists out in the world who used to measure both critical thermal min and max as well as lethal temperatures. And I have an interesting story that I could share with you, but I won't, about some people screaming at a graduate student who was doing thermal biology, and they took issue with the way he was doing it at a scientific meeting. And do any of you know Jacob Marley, former student? He predates you probably. Yeah, okay. Yeah, he was at this meeting and he was sitting in the room and it's like, what the heck are these guys doing? And afterwards, I had to explain to him what the controversy was. So anyway, people can get passionate about how you do these things. So some other things that prior people had talked about in the literature review that they wanted to kind of get some data on. Coles and Bogert were interested in differences between snakes and lizards, and they were also interested in differences between nocturnal organisms and diurnal organisms. So define for me what nocturnal and diurnal mean. If you're nocturnal, what are you? When are you active?
You're active during the nighttime, and then diurnal is active during the day.
Yes, thank you very much. Yeah, so people in the past, before Coles and Bogert came along, thought that nocturnal and diurnal organisms were the same, that there's no reason for them to be different in terms of their body temperatures, their active temperatures. Why would you expect them to be different? Why did Coles and Bogert expect them to be different?
The temperature during the day is different than the temperature at night. Also, there's no sunshine at night. And
yep, temperature during the day is generally cooler than or is warmer than temperature at night. And at night, there's no radiant heat source. And they talk a lot in this paper about the importance of radiant heat sources for animals being able to raise their body temperature. What about the difference between snakes and lizards? What's a fundamental difference between the way a snake locomotes and the way a lizard locomotes? How's snake locomotion different from lizard locomotion that might relate to body temperature?
snakes are actually like on the ground, whereas lizards are at least somewhat elevated because they have legs.
Yeah, so snakes are slithering on the ground, so their entire body is in contact with the ground. As a result, any heating that comes from the substrate, any heat that is found on the ground is going to be transmitted more effectively to a snake's body than to a lizard's body, because the lizard is only coming into contact with the ground at the feet and potentially the hindquarters of the body and the tail. Klauber, who's a guy who worked a lot on snake biology, made the point that for snakes, especially snakes at night, that they can maintain their body temperature at higher temperatures because they're absorbing heat from the substrate. And so one of the things that Coles and Bogert were thinking is that in addition to air temperatures and radiant energy from the sun, the temperature of the substrate that you are on might actually be very important as a source for either warming up by going and sitting on a warm rock, or cooling down by going and pressing your body up against something cool. So how did they go about kind of looking at thermoregulation in this system? What were their methods like? What did they do? Am I beginning to think that you haven't read the paper?
So they had these wire netted cages as a way of like containing the animals, and then they provided a burrow in like the form of an iron pipe below ground as a way of allowing the animals to cool themselves.
How did they get a feel for what the critical thermal maximum might be? So they did these experiments out in nature, but in a nature that's basically fenced in so that the animals can't get away easily. And then you can look at the natural behavior of them in a natural environment that is just altered in terms of kind of hemming the animals in. But that just tells you what they normally would do, you know, moving in and out of sunlight to heat up and cool down and things like that. Then how do you get a handle on what the critical thermal maximum is of a species in that type of an environment? It's on page 278.
They tethered them so that they couldn't like behaviorally regulate, and then also it was easier to take their temperature.
Yeah, so they put them either in a space where there was no shade available and waited for them to lose their locomotor ability, or they tethered them in one of these enclosures where they were inside of shade, and they would watch and see as they struggled to free themselves from being tethered down. They would watch and see when they lost the ability to continue struggling towards the shade, and then would record the temperature at which that occurred. Once again, not a lot of institutional animal care and use committees would agree to such things being done nowadays. But that's how they did that in this case. How did they measure temperature of the animals? They tried two different ways, but they abandoned one of the ways.
They took the temperature of the cloaca, and I think they had banded using the throat.
Yeah, so you can either put a thermocouple into the cloaca of the animal to get an internal body temperature that way, or you can put a thermocouple down the pharynx of the animal Why did they abandon the bucopharyngeal way of doing things in favor of the cloacal way of measuring temperature?
Because one of the ways that the animals would cool themselves was by panting, and so that was dropping the temperature.
Okay, so because, you know, yesterday we talked about respiratory water loss, the loss of water as an animal breathes in and out. That has implications for water balance, but it also has implications for temperature. As you're evaporating that water as you breathe, you are also cooling your body temperature, so that then becomes also a thermoregulatory issue as well. So the temperatures were always somewhat lower in the bucopharyngeal probing of the animal, and they were also more variable, and so they abandoned those ultimately. And ever since then, we still take temperatures of lizards either through a thermocouple or a fast reading thermometer that we insert in the cloaca. But we also nowadays will take temperatures using infrared temperature guns. So that's one way in which new technology has allowed us to be a little less invasive than we used to have to be. So that summarized the methods pretty well. So the latter part of the paper then is them going through species by species for all these lizard and snake species that they worked with in this area of Southern California, and basically talking about what their kind of preferred temperature ranges were, what their critical thermal maximum is, et cetera, et cetera. They also have data on critical thermal min because it would get cold at night. Two of their big questions that they were interested in is, are nocturnal organisms fundamentally different from diurnal organisms in terms of their thermoregulation? So what did they conclude about that, and what kinds of data did they rely on to make that conclusion?
Can you repeat your question one more time? It was about
Go ahead.
Oh, I just wanted another conclusions, please.
So let me go and find where it is in the paper. So at the top of page 271, at the bottom of the first column and the top of the second column, it says there are only 3 truly nocturnal lizards in our desert regions: Phylodactylus, Coleonyx, and Xanthusia, the night lizards. Of these, Klauber reports that only Coleonyx is sufficiently peripatetic, sufficiently available to afford good night collecting. They appear to be active at temperatures as low as 62 degrees and as high as 92 degrees, with a preference for 80 to 84 degrees Fahrenheit. This conforms to the requirements expressed for snakes, and it should be noted in connection with Massauer's statement that there is virtually no difference between the thermal toleration of diurnal or nocturnal snakes and lizards. Nocturnal snakes and this lizard notably avoid temperatures above 33.3 degrees, there being only one record for Coleonyx at this temperature. So one of the things that they are interested in is this difference between diurnal species and nocturnal species in terms of what their preferred temperatures are, and also kind of what their thermal limits are. So they were interested in that question: what did they find, and what organisms did they use to make these conclusions?
I think they found that there was a difference because if you go to figure 3, the means and the ranges for the nocturnal animals are like slightly shifted down compared to the diurnal.
Figure 3. What page is figure 3 on?
293.
The pages with plates on them don't have numbers. Ah. The disadvantage of reading a paper on an iPad. Oh,
And
yeah,
then
okay.
under, yeah.
Yep. Yeah, so you see Crotalus atrox, that's the western diamondback rattlesnake that occurs at least in Kansas, so it's nearby here. Crotalus cerastes is the sidewinder. Arizona elegans is a thing called a night snake. Chionactis is another snake. And then we have Coleonyx and Xanthusia, which are 2 lizards. One of them is a gecko. The other one is literally, it's called a night lizard. And you can see that all of those individuals, all those species have lower body temperature ranges when you look at their normal activity range. What else do you see about those ranges when you look at the mean versus the variation? And I love that this is 1944, and these are clearly hand-drawn figures.
The mean is like slightly shifted up so that their like preferred temperature is at the like higher, closer to the higher limit versus the lower limit.
Yeah, and that's true for both the nocturnal lizards and snakes as well as the diurnal ones. The mean is relatively high within that range, and the long tail isn't a long tail going off to the high side, it's a long tail going off to the low side. But the length of that tail is much longer for nocturnal species than it is for diurnal species. So not only are these nocturnal species active at lower temperatures, but the range of temperatures that they're active at is also much broader than diurnal species. And this is probably due to the fact that there are just more opportunities to raise one's body temperature and regulate their body temperature when you have a source of heat in radiant sunlight, radiant heat coming from the sun that's available to diurnal organisms that is not available to nocturnal organisms. So the nocturnal organisms are kind of forced to be active at lower temperatures because they don't have as many options for raising body temperature. As a result, it wouldn't be surprising if their thermal physiology just evolved to tolerate, and their physiology evolved to be more efficient at cooler temperatures as well, which is why you also see them, having mean active temperature ranges that are lower than their diurnal lizard and snakes that live in the same environment. So, early statements about diurnal and nocturnal organisms being similar just probably aren't true based on Coles and Bogert's work. Another thing that they were interested in looking at was differences between lizards and snakes. So in figure 3, this is the tricky part about figure 3. In figure 3, when you look at the diurnal species, Sarlomalus obesus is the chuckwalla, Dipsosaurus dorsalis is the desert iguana, Crotophytus are leopard lizards and the collared lizard, Scalopris is one of the spining lizards. Uma is one of the spining lizards. Phrynosoma are the horn lizards. Uda is another member of the Phrynosomatidae, the spining lizards. The only snake in that list of diurnal species is Coluber, and it's in the same genus as Coluber constrictor, which Kiara was asking me about yesterday in lab. It's one of the racer species. And so there's not a lot of comparison in the diurnal group looking at comparisons between snakes and lizards, but there are four nocturnal snake species and 2 nocturnal lizard species. Based on this figure, do you think there's much difference between thermal requirements, thermal preferences of snakes and lizards, just based on what's available in the data here? Does it appear that snakes and lizards really differ, or are the main differences between nocturnal and diurnal?
They don't really differ.
It doesn't there's much of a difference between snakes and lizards.
Yeah, snakes and lizards are fairly similar. What really matters is that snake a diurnal lizard, or is that snake a nocturnal snake? So the taxonomic distinction, it doesn't seem to be nearly as important as the activity time of day distinction between these guys. So when you get to the summary and conclusions, reptiles, number 2, contrary to previous reports, nocturnal reptiles not only tolerate but prefer temperatures somewhat lower than those of diurnal reptiles. Provisionally, the difference between mean critical thermal levels for diurnal and nocturnal reptiles may be said to be approximately 6 or 7 degrees centigrade. So that's a pretty big difference in terms of preferences and critical thermal maxima. For example, a 6 to 7 degree centigrade difference is actually pretty big. They mentioned that they were also interested in the importance of body size, and they were also interested in the importance of air temperature being important for thermoregulation versus things like substrate temperature. So what's the advantages and disadvantages of small size versus large body size?
Large bodied size, like they don't dissipate heat as much, so they can stay warmer in like cold environments.
Okay, so the bigger you are, the slower you are to heat up, but also the slower you are to cool down. And so the degree to which your body temperature fluctuates over a long period of time during the day is less. If you're smaller, you gain and lose heat more readily. So if you're cold, you can heat up pretty quickly if you move to a place that is warm. But at the same time, you also have to be a little more careful as a small organism because you do tend to heat up quickly. You have to be a little bit more paying attention to how fast you're heating up, because it's possible for you to overheat quickly because of your small size. So this idea of the size that you are, what that does to your surface area to volume ratio, et cetera, that basically influence rates of heat gain and heat loss. And those happen to be very different for large organisms like Sauromalus obesus, which is a very large lizard, versus Uda stansberriana, which is a fairly small lizard. What was the other thing I was asking about? Size and, substrate. What did they conclude is the importance of substrate in these organisms maintaining body temperature? What's the importance of the substrate?
Like some reptiles can select for positions in or on the soil, like on a rock, where they can heat themselves through direct conduction, or they can bask with the solar heat, things like that.
Yeah, so you can raise your temperature. So there's a whole section in the conclusions about thermotaxis, this idea that depending on what your internal body temperature state is, you will engage in behaviors that either raise your temperature if you're cold or lower your temperature if you're warm. And as far as the substrate is concerned, if you've ever been out on a cool day and then you're standing outside and you want to warm up, if you go and lean up against a brick wall, that brick wall has been gathering heat. And oftentimes, if you lean up against that brick wall, you'll get the radiating heat from that wall that warms up the air that is just adjacent to that wall, and you'll be warmer by kind of basking on that wall than you would be if you were just basking out in the air. And so this transfer of heat from a warm rock to the body of a reptile can be really important for them raising their body temperature. At the same time, if you are a lizard and you're too hot, you can go and dig yourself underneath a rock. And underneath a rock, it's much cooler. And when you press your body up against the underside of the rock and on the soil that is underneath the rock, you then transfer heat once again through conduction to those cooler surfaces, and you can cool yourself down. And so once again, Clobber, who is this snake ecologist, he's a famous rattlesnake ecologist, he didn't find that air temperatures were very well correlated with body temperatures of his rattlesnakes. And he made a bunch of probably incorrect assumptions about what the thermal biology of rattlesnakes were, because he primarily relied upon air temperatures as an indication of what the body temperature of a snake would be, not recognizing that a certain amount of the temperature that a snake will be is actually due to conduction of heat from the substrate that it is slithering on into the body. And so Coles and Bogert basically found that all these different sources of heat, radiant heat from the sunlight, conductive heat coming from the surfaces that the animal is interacting with, evaporative heat loss due to panting and things like this, all of these things come into play in helping a lizard maintain its body temperature, or a snake maintain its body temperature within a particular narrow range. If you go back and look at figure 3, one of the things that you might be surprised at is that these animals are not hampered by the fact that they're ectotherms in maintaining their body temperatures within a certain relatively narrow range of body temperatures. And while they may tolerate colder temperatures, it's clear that they generally prefer warmer temperatures. And once again, the disadvantage of figure 3 is you don't have any idea from that span of temperature ranges how much of that time is spent in those warmer temperatures versus those cooler temperatures, except in that the position of the mean. The position of the mean, if it's way near one tail, that indicates to you that those animals are spending a lot of their time in that range, and that cooler tail is populated by fewer and fewer observations. It would be nice to have seen a distribution of those temperatures. It's just difficult to find that from these data, but you can infer that from where the mean occupies a space within that range. If the mean is near the middle, then you can assume that there's an equal number of observations on both sides. But if the mean is at one end of that range, that indicates that they're hanging out more at those warm temperatures than they are at those cool temperatures. Otherwise, the mean would be lower. So for Monday, we're running out of time here. We are here in chapter 6. If you haven't read chapter 6 already, the part of dealing with thermoregulation, by all means do that. We're going to continue talking about thermoregulation on Monday. Questions? I will see you then.